Dear colleagues, dear academic community members of Babeş-Bolyai University, it is a great pleasure to welcome you to today's session of the Star BBU Institute's Studium Generale series, the interdisciplinary meeting space of teachers, researchers, and students of the Babeş-Bolyai University. By this series, we continue the tradition of quodlibetal questions embodying the spirit of medieval universities, where anything for anyone was discussed within a community of scholars. Today, we explore the intersection of network theory and neuroscience, a field that applies rigorous analytical methods to uncover structural and functional organization of the brain. We are honored to have with us Dr. Mária Ercsey-Ravasz, a distinguished researcher and faculty member of the Babeş-Bolyai University, who has significantly contributed to the field of network science and brain research. Dr. Ercsey-Ravasz completed her PhD as a joint degree in physics and information technology at Babeş-Bolyai University of Cluj and Péter Pázmány Catholic University of Budapest, Hungary. Her career includes a postdoctoral fellowship at the University of Notre Dame and pioneering research at the Babeş-Bolyai University, where she is a tenured faculty member now. Together with Dr. Raúl Mureșan and Vlad Moca, she co-founded the Transylvanian Institute of Neuroscience, where she now serves as a vice president and director of the Network Science Lab. Her work, spanning topics from the exponential distance rule in cortical networks to the analysis of dynamic functional network, has been widely recognized in leading journals such as Nature, Communications, Neuron, and Cerebral Cortex. Today's lecture, entitled Modeling and Analyzing Structural and Functional Brain Networks, examines how network theory is advancing our understanding of the brain's architecture and dynamics. Before inviting Mária to begin, I would like to remind you that the presentation will be followed by a discussion session. Questions can be posted live on the Babeş-Bolyai University's YouTube channel or in person here at the venue. Mária, thank you for joining us today, and we look forward for your presentation.
Thank you very much for the invitation and for your nice presentation. Hello, everyone. So, as presented, my talk will be about, there is a special domain now called network neuroscience. So, somehow an interdisciplinary area between network theory and neuroscience. In the first part, we will talk about structural brain networks. I will present some experimental methods, how these are measured, then how these can be analyzed. I will talk about this exponential distance rule, what we discovered, and based on that, we have built a relatively simple model that explained a lot of topological properties of structural brain networks. And we will also compare them in different species. And in the second part, I will explain what are functional brain networks, the definition, what is the main or the exciting part of analyzing them is that they are very dynamic, they are changing in time, and capturing robust properties is extremely hard. We recently published some new methods that are able to capture robust properties and also enable us to develop biomarkers for different pathological conditions. So first of all, let's think a bit about comparing computer circuits and the brain. So if you think about it in computer circuits, if I'm writing a code on my digital computer, it's enough if I know that it works in discrete time and it's with zeros and ones. So somehow the hardware and the software is quite separated. I'm writing the code, but I don't care where the wiring goes on the chip. I don't even know it, I don't have to see it, I don't care about it. The brain is quite different in this sense because the software is somehow the system itself, the hardware itself. The two cannot be really separated. It's a huge physical network. By physical network, we always mean that in the network you have some nodes and links. And if I'm saying physical network, it means that these nodes and links are placed in physical space and they cannot intersect each other. So like, for example, the neurons would be the nodes of the network and all the axons and dendrites and everything going around would be the links of this huge network. So the so-called connectome is the network of this brain. So it's a huge electrical circuit itself, and everything that happens during our life, it gets somehow encoded in this physical structure, and also the computation itself, what we are thinking, or what we are remembering, is determined by this structure. From the MIT, this Sebastian Seong said once that you are more than your genes, you are your connectome. That's where everything gets somehow encoded. In our brain, the most important part is, of course, played by the cerebral cortex. You also call it the gray matter. Here you can see a slice of an adult macaque monkey's brain. And this gray part is the gray matter, the neocortex, and in the middle, the white matter. That's where axon bundles go through and are connecting different functional areas of the brain. So this white matter is most of it, not only, but most of it, it's formed by these axon bundles connecting areas that are far apart from each other. In a human brain, there are around 86 billion neurons, so it's a huge system. Each neuron has around 10,000 synapses on average. So it's somehow impossible to study a huge network like that. Just for an interesting comparison, they estimate that in the Milky Way there are 200 or 400 billion stars. So it's not even far away from the number of stars in the Milky Way. It's huge, so normally we cannot expect to measure or study directly this network on the level of neurons. So most of the time, yes, actually there are only two species until now where they managed to map this connectome, the whole connectome of a species. For the C. elegans, the little worm, but it has only 300 neurons and 7,000 connections, and it still took like a decade to really map the whole connectome of this little worm. And recently, there was a very nice result. Actually, a whole Nature portfolio appeared with several Nature papers now in October, where they are presenting an interesting database about the Drosophila fireflies. They mapped the whole connectome of this firefly, and there is a lot of information in this database, like 40,000 neurons, and you know all the coordinates. Each of them you can draw separately and look separately at the tree structure of the neurons, like you see here, example one single neuron and the end of its, so it is axonal arbor, you can see it there. But you can do this for all neurons. There is information about 150 million synapses, and all these are organized in 78 larger brain areas, or neuropils, we call them in Drosophila, but functional brain areas in general. But besides these two species, usually we cannot expect to study the structural brain networks on the level of neurons. So we study them on the level of functional areas. For example, in the macaque and also in other species, but they usually have a map of these functional areas. The visual areas are the back of the of our head, in the occipital lobe and so on, the prefrontal lobe. And there are, for example, in this case, in the macaque, there are 91 areas on this map. And as a first step, we want to study how these functional areas are connected. And there are several different experimental methods to study these structural networks, or to measure them, first of all. Diffusion tensor imaging is one of the methods, not so precise. It somehow follows the diffusion of water molecules along the axons, and that's how they try to draw these, you can see these colored axon bundles inside the brain. But it's an indirect method, and has a lot of issues with false positive, false negative connections. There are a lot of arguing about it. They are trying to somehow make it better, because in humans there is no other method available, only DTI. But in animals there are better methods, more precise, and one of them is called retrograde tracing. And actually, this is the method used by the French team of Henry Kennedy. And we started collaborating with them when I was a postdoc in the US. This retrograde tracing means that they have some tracers, like they have some names, fuzz blue and diamino yellow and such things like that. And these tracers, they are injected, let's say, in a target area, and their property is that they get absorbed at the end of the axons. They travel along the axon, and they mark the neuron itself, the soma itself. So after that, after this injection is done, then one can count how many of the neurons were colored by these tracers in each different area. So practically this injection will tell us what are the incoming connections from other areas into this target area. And it is important that there is no secondary labeling, so these tracers don't move on to the next neurons and so on. But we can count the number of neurons, and researchers also estimate the distances these axons can have between the different functional areas. And this way we can build up the structural network. Of course, it's a very costly experimental method. In the macaque, when we started the study, they made injections in 29 areas only. So that meant that we somehow knew a little subgraph of the whole big network. And inside this little subgraph, those areas that were injected, we knew every connection. Let me show you matrix. Here is a figure about how it looks like in the experimental studies when they are making these histological sections of 40 micrometers. And here you can see the neurons that were colored by the injection. And they can count them. This process is already automatized now, and it's doable, but yeah, it's still expensive. And how do we measure? So we have these networks, but it's a weighted network. So if we want to take into account how many of the neurons were counted in different areas. And the weight of these links in this network will be what we call the fraction of labeled neurons. So if the injection was here in the targeted X area, and then we count all the number of neurons in all other extrinsic areas, and we calculate this ratio. So what is the probability practically that an extrinsic labeled neuron is projecting into X from another area Y? So it's a kind of probability of information transfer. And then what we get from the 29 injection is practically a 29 times 91 big weighted matrix. So all the links we get information about. But what we are interested in is this smaller matrix, 29 times 29, because here we know all the information about this little graph. So it's what we call the H-complete subgraph. This is how it looks like when we plot it. It's very dense, but it's strongly heterogeneic in the sense that the weights of the connections cover more than or around 6 orders of magnitude. So there are also a lot of extremely weak connections, and they are also important in these structural networks. Now let's talk about this exponential distance rule and how we discovered it. We were curious what is the relation between the weights of these links and the distances. And if someone plots simply here the logarithm of the weight is plotted as function of the distance, it's very noisy, but you already see a correlation. When you truly see the exponential distance rule, if you count all the colored neurons for a given distance, so practically you want to ask how many neurons do you have with a given axon length, and the probability of having neurons with a given axon length decreases exponentially. So this is what we call the exponential distance rule. This is already independent of these functional areas. Here we added all the neurons at a given distance. We don't look separately at the links between functional areas, just the distribution of axon lengths. And this decay rate it's important. If we measure it here in the experiments, it gives a value. So this is for the macaque. I remember you to tell this was our first study made on the macaque, and it was around 0.18. And then this exponential distance rule comes the question that what can it explain? So if you have this axonal distribution, it drops very fast or exponentially fast. Usually the distance distribution between the areas, it's a simple normal distribution. And we have explained this in a paper, but combining these two already explains that heterogeneous log normal distribution of the weights. This can be shown mathematically. So these two or actually the exponential distance rule itself explains why the weights in this network are so heterogeneous. But we were curious if the topological properties of the network can be explained by this geometrical rule. So is this geometrical rule determining many properties of our brain or not? That was the main question. And we have built a relatively simple so-called exponential distance rule EDR model. This has a one parameter, this lambda parameter, that gives us the decay rate of the exponential distance rule. Here I have written separately that the constant distance rule, but that is practically when the lambda would be 0. So it is somehow part of this, this model, but when lambda is 0, then we say it's a constant distance rule. I just noticed it separately here because we will make comparisons separately for the constant distance rule and the exponential distance rule. And so what we do in this model, imagine that you have the nodes, the functional areas, and the distance matrix is given between them. And now you take this exponential distance rule with a given parameter lambda, and you randomly choose a connection length. So this connection length, imagine that our goal in this model is to drop these axons, drop a lot of axons in this network. So we must follow, we randomly choose a distance, the length of the axon according to this distance rule, and then we place it on a random between an area pair that it has approximately the same distance. We say that it's in the same distance bin, so it's approximately the same distance. And we insert this axon connection in the graph between those areas, and we allow multiple connections. So by this I mean that we allow a lot of axons between two areas. That's how we get the weighted network, so the weights between the areas. And we stop this process only when the binary directed connections in the networks is the same as in the data. So the whole density of the network reaches the same density as we had in the data. Because when comparing the model and data, we need to have the same density, otherwise the comparison would not be fair. So it's a very simple model that gives us a random, spatially embedded, weighted, directed network. And now I will just give you, we studied a lot of properties, but let's just look at one simple example of a network property and how we can define if the model works or not, if can it reproduce the property or not. So for example, here you see M1 will be the number of unidirectional links in the network. So we count all the area pairs that have only one link between them. And the number of bidirectional links where there are both links exist in both directions. So as simple as that. And with the model, we run several, I think 1,000 random models are generated for each lambda parameter. And the average of these measures are plotted as function of this parameter. And this dashed line shows you the value measured in the experimental network. So this figure practically means that the lambda value where the experimental property or the experimental data would be the best reproduced is here around 0.174. And if you remember, I have shown you that we estimated in the experiments the lambda value to be around 0.18, so very, very close. Or I could express this also inversely a bit, that if you look at the value what we measured in the experiment, 0.18, then it would be here, so providing values that are extremely close to what we see in the experiment. So they really reproduce this topological property. And we did this, I won't go into many details, but we did this for many topological properties of the networks. Another simple example, you can imagine this, what we call them triangular motifs in the graphs or networks. So if you take 3 nodes, the links between them can have different types of structure. And there are 16 possibilities here, and we can count how many of these motifs are present in the network, and we can plot the distributions. Anyway, what is important again, that we can define a measure, a difference measure between data and the model, and plotting it as function of this lambda parameter, again we get an optimal value, and again we see that it's 0.18, so exactly what we already measured in the experiments. So fits very well again. And this is true for several topological properties of the network. So besides reproducing many topological properties, the CDR model, also there are some other interesting properties, like this core periphery structure of the structural network. This is somehow shows that there is a very, very dense core, like 92% dense. So only these white links are missing from this little core, and every other link is present, and the periphery has only density 49%. So there is this interesting structure, and this is due to the fact that some very large cliques are formed in this network. A clique is a little subgraph where every link is present, so it's fully connected little subgraph. And the fact that these large cliques are present, again, is explained only by the exponential distance rule model. This is again a histogram. The details are not so important, but only the exponential distance rule at the lambda parameter measured in the experiments, only that one can reach the size of cliques that large as observed in the data. We also looked at some weighted properties of the networks. They call them efficiency measures for information transfer. Now, I left the formula here, but it's not that important. The global efficiency is somehow, you can imagine it as an average conductance between all node pairs. So how efficiently does the information flow through the network? And the local efficiency is a bit more tricky, because asks that if I take out all the connections of a node, will the information transfer still remain efficient or not? And let's stick to this local efficiency measure, because that is the most interesting. And this will be the blue curve on this figure. And what we plot here, that imagine that I'm starting to take out link after link, but starting with the weakest links. And we plot these efficiency measures as function of the density. And what we see, that as I'm taking out the links, this local efficiency shows a large peak. So somehow here, where the density would be already very small, only the strongest links are present here, because I took out almost all links already. This local efficiency shows a large value. And if I compare the data with the exponential distance rule model with that lambda 0.18, then this reproduces that behavior. The constant distance rule, where there is no exponential decay, it doesn't reproduce it at all. So again, actually this this it's a bit harder to understand what happens here, but what this describes that there are many very dense modular structures in this network, and this is known to be true for the brain. Now, just some interesting questions came after this study, that does this EDR hold in other species, and what changed during evolution? Well, first of all, we know that during evolution what changed was the size of the brain. And not only the size increased, so the volume of the brain increased during evolution, but the surface of the brain increased even much faster. That is the reason why this, what we call gyrification appeared. So the surface of the cortex got very, gyrification. You can see on this figure, for example, a slice from the mouse brain, the rat brain, macaque, and the human. It gets even, so more and more, this kind of fractal structure starts to appear, this cortical gyrification, they call it. And in 2014, there were some experimental studies in the mouse, again using track tracing, but anterograde tracing, where these fluorescent molecules follow the opposite direction. So they get absorbed at the dendrites and get released at the end of the axons. So the method is not as precise because one cannot count precisely the number of neurons. But anyway, the binary connectivity matrices in the mouse could be also measured. And actually in the same year, two different groups measured these in the mouse, and two data sets appeared. And because they are not so precise, both groups, I think they reported the connections that they really thought to be trustable. But the result was that there was a lot of difference between the two results. I mean, the two connectivity matrices, even if they use the same map and the same segmentation atlas. There were a lot of differences. So when we wanted to study, we were thinking what to do. We decided to summarize the two matrices. So we said that, okay, if one group said that this is trustable, then probably it is. And indeed, by summarizing the two matrices, probably it seemed weird to researchers, but these networks are really dense. What is important that the weights are extremely heterogeneous. So even if the network density is almost 90%, but there are some very, very weak links, and there are some much stronger links. And that was important. I don't go in the details of this. I have shown you the similar figures in the case of the macaque. Again, we had analyzed several different network properties. And at the beginning in the mouse, we didn't know what was the experimental value of this lambda decay rate of the exponential distance rule. So we tried to estimate it based on the model. So what would be the interval where it sort of estimates most of these properties? And we estimated, I don't know, this interval, and then later on, the French group made some retrograde tracing experiments in the mouse, and we got a very precise measurement for the exponential distance rule, the decay rate of 0.78, that matched quite well with what we expected. So it seems that this EDR model also worked in the mouse. And we made also some comparison between mouse and macaque. And what really changed during evolution is that if we would scale down the brain size of the macaque to the same size of the mouse, for example, so we divide there on the x-axis you would see that we divide the distances by the average of the distances, then it's practically everything is very similar. Even if the distribution of the motifs is similar, or certain similarity measures as function of distance, they look similar. So it's really, it seems that the scaling is what changed during evolution. It's interesting to mention that even the EDR is true even inside the gray matter. So what we were talking about until now was the structural, I mean the exon bundles crossing the white matter and connecting different brain areas. But it is true also in the gray matter, only with different decay rate, but approximately the same decay rate in mouse and macaque, for example. And let's mention shortly the Drosophila, because it's a very exciting new data set. And it's also much more complicated to analyze because there is no separate gray matter and white matter. And even the structure of the neurons, it's very weird compared to what we have in mammals. So the soma is usually separated and it's outside somewhere on the periphery of the brain. And then a primary neurite comes inside the brain, and there it separates the axon and the dendrite to the dendrite and the axonal end. And here we wanted to test again the exponential distance rule and actually to measure it precisely, the decay rate. And in this data set, we can separately draw this structure for each neuron. So we measured this axonal length, but we also measured the so-called Euclidean axon length for each neuron. And this was important because between the functional areas in the Drosophila, we are provided only with Euclidean distances. So we somehow, when we build the same type of model as I already described, we do the same thing, but we need to estimate what is the probability of the axon to project to a certain Euclidean distance. So we need to separately calculate this lambda value for the Euclidean distances. It turned out to be around 33 inverse millimeter. And the model worked again very nicely. I mean, this data set also, they built the neuropil projectome, so the connectivity matrix on the level of these 78 functional areas has been presented in these publications. And we analyzed these. Again, all the network properties were quite nicely reproduced. So actually, I would like to talk here about one extremely interesting property that cannot be reproduced by the model, and why. So this was the symmetry of bidirectional connections. So imagine two nodes which are connected in both directions, and we calculate a so-called asymmetry measure between the weights. So of course this EDR model is based on the distance matrix that is symmetric. So you cannot expect this model to produce you very asymmetric connections. It will randomly put the direction of the axons or in that direction or in that direction, but it's completely random. So it's expected that this EDR model is somehow more symmetric. But why are the experimental data sets, why are they showing so much asymmetry? For example, here, this is the data, the gray columns. And for example, the EDR model is the black one with the same lambda as estimated. And it looks completely different. But on the right side, if we plot this only for the links that are connecting functional areas, so the same functional area from the left and the right side. So we call them the homotopic bidirectional links. So for example, I don't know here the names because they are so different from the mammals, but the visual area from the left and the visual area from the right. Now, these are much more symmetric connections, and they are reproduced by the model. So this interesting property with the asymmetries is related somehow to the functional hierarchy of the areas, because it somehow estimates that, okay, for example, input comes in our visual cortex, it's moving further on to be processed, but much less information, some information does come back as a feedback, but much less information is coming back, and this is what causes these strong asymmetries in the brain, the functional hierarchy. We even made an estimation of this functional hierarchical ranking in this our last paper, which is under review now. Okay, so just a few conclusions about the structural brain networks, that we can see that this applied null model plays a crucial role, and it is important that it incorporates unavoidable geometrical properties of these physical networks, because we must know what is the property of the brain that is really due to its physical structure, to the geometry. It cannot be avoided. And if we apply these models, these EDR models as null models, then we can more easily discover relevant and biologically interesting functional properties. And it's quite amazing that it works in insects, rodents, and primates as well. So it's really general and should be used in future studies. Now, just shortly about functional brain networks, because I see I'm running out of time. So we talked already about functional areas and the network between them, but when we say functional brain networks, we mean that now we are interested in the activity of brain areas and the correlation between them. So the connections now in the network won't be structural in the sense that where do the axons go, but we build a kind of correlation matrix, and we are interested in how well the activities are correlated. The problem is that it's very dynamic. These are changing in time, even during resting state. If you are just lying with your eyes closed, even then these are changing in time. So the analysis is really complicated, and the results are usually hard to reproduce. And these are measured with fMRI, EEG, MEG, COG, and so on. So just a few words about the two main, mostly known methods, electroencephalography, so the EEG. This is an image from the teens lab and their EEG processor. So what is really that you put on a cap with, I don't know, usually 128 little electrodes, and they measure the electrical signals through the scalp, and they look like this in time, very noisy and so on, fluctuating. You somehow try to measure correlations between the signals of each pair of electrodes. Or in case of fMRI, so the functional magnetic resonance imaging, this shows the bold contrast, so the blood oxygen level dependent contrast. This is useful because if a functional area is more active, then the oxygen level of the blood is higher. So somehow it correlates with the activity of that area. It's not as, the relation is not as direct as in case of EEG, but still we get information about where the activity is higher and stronger. In case of fMRI, we get information about little three-dimensional voxels. These are usually, the functional areas are identified, and we get information about these a general value of the activity in a functional area, or these regions of interest, they also say it. And the only problem is that the temporal resolution is much slower. So just shortly, this will be really brief because there are a lot of mathematical details here, but this new methodology, what we recently published about functional networks in cell systems, we tried to, two main ideas were introduced here, that the time lags in brain communication are important for constructing these functional networks. And when analyzing these functional networks, we must look at the statistical distributions of the network properties and not the averages, as was done in many, many previous studies. And you will see why immediately. So we have a lot of experimental data when we published this paper. There were rats with alcoholic conditions and control group. There were also humans, alcoholics, and a control group, and even a marmoset, a healthy control group. So we tested this method even across different species. Now, just a simple image to give you an idea how we build the functional network. So imagine that this would be the signal from two different or electrode, if it's EEG, or region of interest, if it's fMRI, whatever, but two signals. And I want to somehow calculate the correlation between them. Now, we do this by building a cross-correlogram. So there is this time offset. Relatively, it must be shorter than the signal itself. And then we apply this time offset, and we calculate the correlation in this overlapping period between the two. And here you see the correlation value as function of the time offset. Now, if the time offset is 0, then this would be simply the Pearson correlation coefficient. Here on the right, you see the correlation for each link in the network as function of the time offset. So in case of the Pearson, each time offset is 0. And the distribution looks like it's almost always positive. Now, what we are doing differently is that we always take the maximum value in this cross-correlogram, allowing these time lags to exist. So we don't take it at 0. This is important because it takes time for the signals to reach from one area to another, so this shouldn't be neglected. These are very important. In fact, the whole distribution of these network the weights, the correlation weights, it gets completely different. It's a bimodal distribution now, so it looks differently. And here I am showing you as an example how dynamically changing are these functional networks. For a rat, for example, in an epoch, so a time session 1 or 3 or 5, but during the same fMRI session, in fact, but we cut it in 5 periods, and if you plot the network, looks completely different. So red links are positive, blue ones are negative. And if you would take the average, you would get very, very different results. And I would try to explain this with a very simple example. Probably most of you know about this simple dynamical system, the Lorentz dynamics, which has this butterfly-shaped attractor. Now imagine that you take small samples from these dynamics. So the blue one is the attractor itself, and you would take short red samples from the dynamics. Now, if you take after that the average of these sampled values, you would get a value somewhere here in the middle, which is not even on the attractor. So it's not realistic. It's never, the system will be never close to this value of average. But if you look at the distribution of these coordinates, for example, let's say the distribution of x, the small samples, the distributions already approximates well the distribution of of the coordinates obtained by looking at the attractor itself. So this is just simply the idea that when looking at properties, we must look at the distributions and not at the averages. And you can imagine that in the brain we have much more variables, not 3 like in this simple example, so it's even more complicated. But looking at distributions can give us much more information. Here, for example, in the rats, so we had 18 rats in one group. Each of them had 5 different epochs in the session, and we have certain links that are always strong and positive. The red means strong and positive. And there are certain links that are somehow bimodular, changing all the time, sometimes positive, sometimes negative. So this strong positive backbone, we call it, is always present, and some links are changing a lot in time. So what I will show you here on the next figure is what we say the edge weight distribution. Now here we have a lot of edges in this network, like 3,000, I don't know. So one little row you can't see even as a row, but even if we zoom in, so one row would be the distribution of an edge weight between these 90 values. Here are 90 values. So for the 18 animals and 5 epochs, 90 different values, and I will plot for each link the distribution of these values. But the distribution will somehow coded by the color to be more easy to see. So one row is, for example, the strongest link will be here at the top, and it's always positive and extremely strong. And then we order them according to their strengths. But what is truly amazing that we keep the ordering, so we make the ordering based on group 1 with 18 rats, and we keep the ordering in the other group where we have different rats, so different individuals, and at 2 different time points, 2 different scanning, but this robust pattern is preserved. And this was really what surprised experimental neuroscientists, that usually in fMRI it's hard to obtain results that are so robust. Yes, I skip this. Okay. And another measure what we applied here and was useful to characterize the functional areas in these results. It's a kind of centrality measure. We call it node distance distribution, but again, it's always distribution. It's much more useful to work with distributions and not average values. For example, if I have a little node here, I calculate the shortest path to each other node, and I just plot the distribution of these distances. So this is how you can imagine this distribution. And again, we have a figure where each row shows you the node distance distribution for one functional area. S1 left or S1 right, and so on. And we order it according to the first group, and we preserve the ordering. And then we can also compare these patterns and find out where the most changing occurs or so on. And yes, this is the ranking of the areas based on this centrality, and it produces very symmetric figures in the brain, so it's very realistic from this point of view too. And this is just a short example how we obtained information about what the alcoholic condition changes in the brain. So for example, here in the middle, you see the control group of the rats, so they did not get any alcohol, and they were scanned at 2 different time points of 30 days. And here the differences are quite so This delta, cliff delta value, it just compares the two distributions. When you see here white, it means insignificant changes. The green is a very small change, and the orange is medium. So between these, in this control group, the changes are really, really small. When we have this G1 group, these rats had alcohol in those 30 days between the two scannings. And here the differences are already larger, usually small and medium. And this is a human study, a control group and the patients. Here the difference is that you cannot have the same rats in, I mean, before, you cannot have the same. So these are different individuals. Here in the G1 group of the rats, you have the same individuals. In humans, you had to get different controls, and patients, there are different individuals. So these differences are even larger because of that. But what is important here, what I'm trying to say, that one can identify areas where the changes are the most largest. I skip this. The conclusion here in general, and you can see this here, in case of humans, that these distributions are shifted a bit to the right and wider. This is caused by the fact that these correlations get weaker by alcohol. So after drinking alcohol, somehow the correlations get weaker, and the centralities, these distances, what we measure in the networks, get larger. And also the efficiency gets smaller. So this is what is shown here, but let's skip this. So we can identify areas that show the largest differences with this method. For the example, the amygdala, that was known to be very important when talking about drugs and alcohol and this sort of behavior and so on. So as a conclusion, in this second study related to functional networks, it was very important to take into account the time lags between activities, and this idea of studying the statistical distributions of the functional network properties and not averages. And it gives, yes, we saw this backbone of functional connections, and it gives opportunity to identify sensitive biomarkers. We have ongoing projects related to consciousness disorders and amblyopia, and many different data sets are promised. We are starting other projects related to autism and depression and things like that. So it's a really promising method that it can also be generalized. I was talking now about fMRI data, but it can be used, and we already applied it on EEG data as well. Okay, thank you very much. It was a bit long. Thank you very much. And if you have questions.
Thank you, Maria, for this interesting presentation. And now we go forward to questions. If there are any questions, comments related to the presented topic, then feel free to ask.
Did you thought about the fact that you could try this experiment with people of, I don't know, different ages, and you can see how they react?
That would also be very interesting, just hard to implement. But yes, so we should, on the long term, the most interesting part would be to look at individuals at different ages. That's why I'm saying it would be hard to implement, because if you look at different individuals, maybe it's harder. With fMRI, it might be more, more robust. as we saw until now, when we apply the method on EEG, it's much more sensitive to individuals because the EEG cap, as you put it on the head, it's not as precise when while in fMRI they can identify those functional areas much more precisely. But yes, it would be interesting to take groups of individuals at different ages and look how this functional network is different or it develops. It would certainly be interesting. It's a good idea. Yeah?
What sort of mathematical tools do you typically use? You skipped at some point the mathematical part in one of the papers. So do you use dynamical systems, or it's mostly based on distributions like coming from probability?
Well, it's one of them when we are extracting the functional networks. There I have showed you that we use that cross-correlogram, but actually there is another step to it, the scale cross-correlation algorithm that was published by Vlad-Moka and others. So here it was. So I explained it until here. There is another step. It's just this scaled cross-correlation, scaled correlation analysis is just able to emphasize the fast correlation. So this is another mathematical part. Then other mathematical parts are related to how we calculate all these graph properties. So I did not go into details, but for example, how the distances are calculated. So we have that correlation weight, but the distance, so the stronger the correlation, it means that the distance is somehow smaller, right? So actually we define the length of a link as minus the logarithm of that correlation weight. And then if you add up these lengths of the links along a path, it's really gives you back the probability of information transfer. This is also shown in the method section of the paper. So this is another part that is a bit more mathematical. Then it's about, I think near that it's not that complicated. I mean, we're really just working with distributions and not averaging, and some statistical measures and so on. But it's not that complicated, just yeah, we actually publishing, it's under review now, a protocol paper which gives you instructions. So we made the codes available on GitHub, and that paper will give very precise instructions on how to use them.
I was actually wondering whether there would be any benefits in considering systems of ordinary differential equations, for instance. You also mentioned Lorentz, rather than just correlations between, because...
Mm-hmm. Well, I mentioned the Lorentz only as an example to give you that average is not a good idea. But
the mathematical part, not from neuroscience, that's why I'm wondering what type of models are used to model.
But if you, so you are thinking probably about these models of these neuronal models or larger brain models. Yes, probably there we could also apply similar methods. So if you have, for example, a model with a huge system of differential equations, you practically can have these signals extracted also from there and apply the method in a similar way. Thanks.
I also have a question. As I understood right now, you have presented some mathematical models through which, I don't know, the brain was viewed as a complex network of networks, something like this. And my question is, I think that is because physicists like to keep our models as simple as possible. And the question is, what do you think such kind of mathematical models are some limitations, or in the future we will I don't know, have a point at which we say that we know anything we can about the brain through that models, and we have to step forward to, I don't know, to more complex modelings, or what is the future?
Well, asking about the future, that is a very hard question. We don't know the future, we just know that there is still a lot to learn about the brain. But when you say that complex network of networks, what did you mean exactly? So
I think I mean that we have this functional part which are also composed by in some sense by networks.
So you can imagine these functional networks. It's like a network changing in time, and that's what makes it really complex. So it's like you would imagine a network, and the weights of the links are practically changing in time. And you somehow, actually, we don't have mathematical methods that are precise for these types of systems, I think. So probably better mathematical methods would be needed and would be welcome for these studies.
And what about the computational efficiency? I think you have to make compromises to have between resolution, data complexity, and
yes, actually, there are larger and larger data sets coming now. For example, in functional networks nowadays, there are other methods of extracting data from the brain. They sometimes insert these little. I have thing. Can I show a hidden slide? I think I put it here at the end. No, it's against the. Just a moment. Yes, from current slide. So this is in Hungarian, sorry, but the picture, what it matters, that somehow they insert these little multi-electrodes in the brain, for example, in the case of the mouse, but they even do it in case of humans when it's needed for medical purpose. And nowadays, these multi-electrodes have not only 4 or 8 or chips, but many, many, many. I mean, you get a data set that it's even hard to store somewhere, not to analyze. So really, the computational limit is a problem in those cases. And even in these structural brain networks like the Drosophila, I mean, there are so many connections. We studied this network only on the level of the larger brain areas, but it's the network, and we really would be curious to continue the study on the level of neurons, because everything is present and known. So why not build a model that tries to model the level, the connectome itself on the level of the neurons? So yes, computational limits are a problem usually in these research areas. So processing all this data, storing it, yes, analyzing, modeling, it's usually very costly.
Thank you. Okay, I was looking, and there are no questions at the YouTube channel. So actually, I would like to thank you, Maria, for this enlightening presentation that I hope provided us with a deeper understanding of the brain through the lens of the network theory. And we are also grateful for the opportunity to delve into such interesting topics in this Studium Generale series. And I would like also to thank you for all the Babeş-Bolyai University academic community members who joined us today, and we are looking forward to meet you at our future presentations within the Studium Generale series of the Star BBU Institute, transmitted live from here, from the Taylor Bastion of Cluj. Thank you.
Thank you very much for coming, and Merry Christmas, everyone.